Is there wine? That's funny. Good to see you. I'm surprised you came.
Ik ben right now outside surf around going to this spider talk.
Good.
Usually wait like 5 minutes after the time.
It's fine.
I know you said we told you to come early.
Don't worry about it. Yeah, you really don't need to apologize. It's not an issue. If technology didn't work, then that's crazy, isn't it?
Amazing.
I don't predict that ever.
No. I have a friend who would never go near. I mean, I guess it's one of those strong phobias.
It's a divisive topic.
I think I'll stay.
You'll be all right.
It's the beard, I think.
Yeah.
I'm also involved in the Red Pan Museum's organizing committee for this lecture series, the Cutting Edge Lecture Series. So I officially welcome you to the museum tonight. Tonight, the students who are running the crowdfunding campaign for this series, they are hosting tonight. They'll be introducing the speaker, they'll be introducing their campaign, and after the lecture, they have set up a Sunday bar. Spiders in the Arctic, ice cream, perfect fit. In fact, the poster, even the way the artist did the poster, it was the Red Pan Museum covered in snow, and there's a red spider on top of the Red Pan Museum roof. It looked just like a sundae on top of the museum. So we're happy to meet you in the dairy bar. It's room 106 with a contribution, $5, suggested for 3 scoops, lots of good toppings, 1 scoop, $2. We accept checks. I even have a credit card if you want to really go whole hog on that. So now I will leave it to Christine to introduce the crowdfunding campaign.
Hi everyone, I'm with the crowdfunding campaign that supports the Cutting Edge Lecture Series. So this year we're carrying on our tradition of raising money to help fund the Cutting Edge Lecture Series, which has been going on at McGill for 13 years now. So if you'd like to help support our campaign, please join us for ice cream. Or if you'd like to make a donation at any other time, you can find us on the Seeds of Change website. So just Google that and you'll find us. Thank you.
So Welcome everyone to the third lecture of 2016 Cutting Edge Lecture Series. Thank you very much for being here. My name is Val. I'm also a student fundraising for the museum and for this amazing series. And tonight I have the pleasure to introduce my speaker, Dr. Buddle. So Dr. Buddle has been working at McGill since 2002 in the Department of Natural Resources Sciences at McGill's Buddle campus. His research is focused on the biodiversity and population ecology and community ecology of Arctic spiders and insects in the Arctic. It has been long involved with academic programs and administration in the Faculty of Agricultural and Environmental Sciences, most recently teaching field biology and ecology in the Environmental Biology program and holding the position as Associate Dean, Student Affairs. And Chris was appointed Dean of Students last August for a 5-year term. So he's a recipient of many awards, including Excellence in Entomology in Canada, awarded by Entomological Society of Canada, as well as the Manual Campus Award for Teaching Excellence and the Desire to Learn Teaching Innovation Award, awarded by the Society for Teaching and Learning in Higher Education. So this is what you can read on Dr. Buddle's CV. For me, Chris is first and foremost one of the first faces that welcomed me at McGill in my first year when I arrived. So the first class I've taken at McGill was field biology and ecology with Chris when he was an associate professor in 2014. So when I arrived at McGill campus, I was a non-native speaker. I didn't know anything about McGill, the way science is done in America, done in Canada, North America, I meant. I found in Chris a mentor and by far the best teacher I had at McGill, at least in my first year. And the continued source of inspiration and help through the posts he made on his blog, the help he conveyed through his grad students that was very inspiring also. And finally, an example of very good public speaking skills. So that's when I was asked to become a speaker for the series. I certainly thought okay. So Dr. Buddle, thank you very much for coming here tonight and showing us some science. Something you told me you were missing in your daily duty of Dean of Science students. We're all very happy to have you here. So ladies and gentlemen, please welcome Dr. Chris Buddle.
Thanks, Val. I'm a little nervous now. Thank you. It's great to see some students here that I've taught before, and I love teaching, so the irony is not lost on me that I'm not teaching this year as Dean of Students. So, and yes, I'm missing the science. Real pleasure to come and talk about some of the work we've been doing in the Arctic. I can't believe this many people are here because usually when something is about spiders, it tends to be a fairly small audience, but very devoted. So this is great news. I think this is a great moment for arachnology in Montreal. Yeah, I talk about other stuff too, but arachnids are certainly my passion. So I'm gonna tell stories today. I think stories are more interesting than science without the story. Take you through some of the research we've been doing and talk a little bit about some broader philosophical ideas around the way we think about science in the field. Okay, as Val mentioned, one of the courses I teach is a field biology class, which has really changed the way I think about teaching and really made me reflect on how we interact with nature. So I'm going to try to talk a little bit about that. But the story today really starts with something called the Northern Insect Survey. Back in the Cold War era, there was a distant early warning sites that were all along the Arctic that were essentially set up by the Department of Defense in Canada so that we could warn North when the Russians flew over. The dew line, it's called. As part of that, there was concern about the effect of biting flies on military personnel in the Arctic. That makes some sense. If any of you have been to the Arctic, there are biting flies up there, and a lot of them, okay? So there was some, it's not like other people in the Arctic know how to deal with biting flies, but nevertheless, entomologists were sent north. They went to this many spots. This is unbelievable. A vast area across northern Canada, Alaska, Greenland. Entomologists, those people that study insects, landed in those spots, funded by the Department of Defense, to collect insects. That's a lot of sampling. Canada's very big, okay? And the expense it takes to get to each of those spots was immense. Why am I starting with that talk? Well, because we know that the Arctic is changing, okay? It's a very interesting place for so many reasons, but it's very alarming because the rate at which things like climate change is happening in the north is very, very rapid. This is a map from 2010 is when it ends, actually, but the red spots there, dark red means bad. That's where the effects of changing climates are going to be felt most significantly in our most northern regions. And it's not just climate change. Climate change affects things like the Northwest Passage, which increases traffic through the north as well. And there's been incredible development in places like Iqaluit and other northern communities where populations were 2,000-3,000 people for a long, long time. Now Iqaluit, I think, is over 8,000 or 9,000 people. There's been an incredible amount of growth and development. I've not even mentioned mining exploration and ecotourism. There's so much influence happening in the north that it is a fragile Arctic. Okay? And that's part of the context when we think about that northern insect survey done in the late 40s. Well, there's an opportunity here to think about where we are today. I was involved with a group out of Iceland, the Conservation of Arctic Flora and Fauna. They're an international group, pan-Arctic, trying to develop what we call ecological monitoring protocols. Ecological monitoring or environmental monitoring has different words. It's essentially to try to benchmark where we are with our biodiversity. And our biodiversity is the diversity of the different species. That's how I'm defining it here. So I was part of a group that looked at trying to develop ways that we standardize protocols in the north so we can properly understand what's in the north so that as we continue to move forward, we can understand if things are changing. Okay, that's a key piece. Okay? We know that it's changing. We know there's a lot of influences in the Arctic, but is the flora and fauna, are the animals and plants, are they being affected? And that becomes very important. We can talk about countless examples. Think about pollinators and flowers. If there's not a sync between when pollinators are pollinating flowers, that's a problem. Pollination won't occur. We think about wildlife species that depend on insects. I always give an example of a lot of the breeding birds that first show up in the Arctic, they feed on spiders primarily. So we don't think of that often, right? Birds eating spiders, but it's a really important food source. So if there's a decoupling or a change in when snowmelt happens relative to when birds arrive in the north, it's a big deal. So you can guess where I'm going with this. We got a whole bunch of money to go back to the spots that are in blue there. We did a project called the Northern Biodiversity Program. We didn't get enough money to go everywhere. That would take a long time and way more resources than anyone would give us. But we were able to go back to some sites over a 2-year field season and start to think about whether or not we're seeing change over time. I'll tell you the bad news right away is that although we like to think that the entomologists from the 1940s were extremely careful in their approaches to sampling, it maybe wasn't as good as we would have liked. Okay? Ideally, there would have been a standardized way that we could then compare back in time and really understand change. So, and it was tough to be able to document change because of the way that the entomologists looked for stuff in the 40s and 50s relative to what our expectations or how we approached the question in 2010. At the very end of the talk, I will talk about one piece that we found in terms of that changing climate, but in general, we had to refocus and think about not so much where we were historically, but where we are now in the Arctic. And that's what I'm going to talk about, some stories relative to some of the sampling we did over these 2 years and some of the stories that evolved from that. So just briefly, to talk about that Northern Biodiversity Program, the landscape in the Arctic, how many people have been to the Arctic? 2, 3. Yeah, not very many. One thing that surprised me, and maybe those that have been there would agree, I imagined it before I went to the Arctic as being homogeneous, sort of a uniform kind of place with not a lot of variation in different types of habitats and kind of all the same. Big, open, wide open, cold, and the same. And it's just not that way. It's so diverse. And even if you look at this photo, you can see all these sort of micro variations in habitats that suggest far more, we call it heterogeneity or variation or different kinds of habitats, even in a landscape that we might think of as being sort of flat and kind of uniform. And this actually proves a challenge for an entomologist because our idea was, well, it's going to be easy. We'll go up there and sample, but we actually had to use all kinds of different techniques and try to capture that variation in the different habitats as part of our sampling programs. I'm not going to talk about all these different techniques, but I will assure you that these are legitimate techniques for collecting bugs in the Arctic. Okay. They really are. And we didn't only collect on land, we also collected in the water. It was a massive undertaking, and there were 4 different universities involved. We trained a lot of graduate students. I got to go to some of the sites. I let the grad students go to the most exciting ones. I regret that a little bit. But I don't know. I'll get back there. So what do we find? Okay, if you're scared of spiders, sorry, I should have warned you. There's a big spider picture coming up here. Because as an arachnologist, my lab was involved with looking at the spiders that we collected from the different techniques I just showed you. And just to give you a snapshot of the data, we collected about close to 30,000 spiders, which is fine. 14 different families, that's a fairly high level of classification. But 306 different species. That's a lot of different kinds of spiders. And the question I always get when I study spiders in the Arctic is, are there spiders in the Arctic? Yeah. Absolutely. And there's a lot of different kinds. 131 species were new territorial provincial records. So these weren't new to science. We have one species we think is new to science. 131, that means that although science had known the name of that species before, no one had collected and identified it from provinces or territories before. That just shows, here we are, that was 2010, 2011. Spider taxonomy, the process of describing spider species, it's fairly mature, it's fairly well known, yet a huge chunk of planet, right, the Arctic in Canada, for the vast majority of that space, people hadn't collected any of the species there before. That's very reflective of how much work we have to do as biologists, okay? It's a big undertaking to try to understand our biodiversity. Can you see the little spider up there? That's a mature adult spider. Most spiders are small. I don't know if that's a comfort to you or not. It may not be, because in your home you've got a lot of spiders that are small and you're just not seeing them. Of the 306, yeah, I'm sorry. Of the 306 species, I think 169 of those species, if I got the number correct, are less than 4 millimeters in size. So most spiders, especially in the north, there's very high diversity of small spiders in the north. And they live down in the tundra. They tuck in there and hide in the tundra. I thought I should show some data. Val, you know how proud I am of good slides. Can you read all those names at the top there? This is all the spider species, but I just want to illustrate that the green bar there shows the number of species found in the North Boreal, and the blue is the subarctic, and then 37 species are found in the high Arctic. So that's kind of the distribution of all the species. What I found interesting, maybe you don't find this interesting, I find this interesting. 9 species are found in all 3 of these major biomes. That represents an incredible plasticity for a species to be able to live in Moosonee as well as in Lake Hazen at the top of Ellesmere Island. That's pretty interesting, that degree of plasticity and adaptability to different climates. It's kind of cold everywhere in the winter there, but it's a whole lot colder in the high Arctic and a whole lot more desolate, whole different vegetation structure. 9 species. I was surprised by that. So more sciencey stuff. To take away from this, Those dots on the left represent the sites in the high Arctic. And this is a quantitative way to illustrate the whole community. That we describe all the different spiders, how many of each species are found in the different biomes. The point I want to make with this is that the Arctic is different. Okay? There's overlap between the North Boreal and the subarctic, but the Arctic communities were special. They were definitely different. Different species, different numbers of those species. So we have to pay attention to them. It's not only is it not a desolate, empty landscape, it has a fauna of spiders that's different as well. I also had a graduate student, Crystal, who worked on beetles. And she collected a bunch of beetles, 464 species, and 18 functional groups. So she looked at how the beetles lived their life. Were they predators? Did they feed on plants? And describe that functionality of the beetles as part of this major project. There were lots of beetle species found in the south and in the northern boreal region. Only a couple species at the very top in Lake Hazen, but they are everywhere. And this is the same kind of, don't worry, the sciencey part, the boring sciencey part will be over soon. Stay with me. By number of species on the top, the high Arctic sites are separate again. The bottom graph shows the ecological function of beetles. The functions provided by beetles in the high Arctic as a whole are different than what we see further south. There's a different. They're hypothetical gradients. They don't represent anything. In theory, they represent some environmental gradient, but it's a best fit model, so we don't have a specific environmental gradient along there. All it represents is the relative difference between the points becomes important in this analysis. So it represents an explanation of the variation. It's not an actual unit per se. Okay, so that may or may not have helped either. All right, so where are we? Let's pause here. What have I talked about so far? We've talked about the Arctic. It's changing. There was this project because people went up there in the 40s and 50s and collected stuff. We went back in the 2010, 2011, collected more stuff, and I've just illustrated a bunch of patterns, right? Things we found, stuff we found where. That may be interesting, but I think the somewhat more important question is, why do we see these patterns? So it's one thing to document that we see these patterns, but I'm immensely curious to ask, why do these patterns happen the way they do? And hopefully some of you in the room have the same curiosity. Some people would be very satisfied with seeing that map with the different points on it and the number of species. But why? Why do we see those patterns? And this is actually a deeper level ecological question, and we need an answer to this. We have to understand the reason behind the patterns if we're going to be faced with changing climates. Okay? That's the argument I'm making. So let's talk about squirrels. Well, they live in trees, and they collect nuts. So says Emma. That's a pretty good observation. Okay? That's a grade 4 observation, I believe. Squirrels live in trees and collect nuts. What is that? That's a depiction of natural history through the eyes of Emma in grade 4. When I say natural history, what comes to you? Well, you're sitting in the Redpath Museum, so you're probably thinking of this. You go to the Natural History Museum in London, or the Canadian Museum of Nature. We think of dead stuff presented for the public. Okay? That's how we often think of natural history. But natural history is more than that. Natural history is a foundational piece related to how we observe nature. Okay? Scientific natural history, telling the epics of nature. Scientific natural history, which addresses all biological aspects of individual species, one species at a time, and then compares many together, is important to science as any of the umbrella biological disciplines. So there's an argument, and there's been a lot of arguments in recent years, that we need to tell the stories of species. They have a story to tell. We just don't know it for most species. And if we're going to strive to understand a pattern, we have to begin to pick away and understand the different species. That's why you start with a kid looking at a squirrel. Squirrels and trees, they eat nuts. Let's advance that. Natural history is a foundational science, and it has a place in today's science and society. So I'm going to tell you some natural history stories related to the Arctic patterns that I've already showed you, to try to make a case and reflect on whether or not we can dig below the surface of those patterns and learn from the species in these systems. And they can tell us some stories that will help inform us more broadly. These are pretty nice looking beetles. Not my picture, but the colors are accurate. Okay. Henri Goulet is an entomologist in Ottawa, and he allows us to use these pictures. It's morphological color variation in the same species. I mentioned my student Crystal, who was looking at beetles, and she spent an entire summer in Kuguktuk, which is in the westernmost town in Nunavut on the mainland. Okay? And she spent a whole summer there trying to understand those beetle communities, not just as a quick drop-in to sample, but actually spend some time there and learn more about the beetles that are there. And then she noticed this. Doesn't look very comfortable. It looks wrong. And it's not one species, it's a hairworm that's emerging from the anus of the beetle. Okay? So as Crystal was sorting through the beetles, so she spent time collecting the beetles and got back to the lab and did exactly that. She went, huh? What's going on there? And then she went, huh? A whole lot more. Oh, sorry. You thought spiders were bad. Just wait till I get to show you some other stuff here. These hairworms were emerging from a whole lot of beetles in the Arctic. Those are some of the sites. She documented hairworm beetle associations almost 1,000 kilometers further north than had ever been documented before. All of these are new records for provinces and territories. And it's an unknown species of hairworm, and I'll tell you the story of that hairworm next. But parasitism rates, so the number of beetles infected with these hairworms, was ranged from 13 to 21% based on the sample. You look at a sample and see what percentage had parasites. It ranged from 13 to 21. Beetles were infected regardless of sex, body size, or species, so the hairworm didn't seem to differentiate based on those parameters. But more infected beetles were found in wet habitats. This was the story that Crystal told. Here's the story of the hairworm. The adult hairworm to the right, that hairwormy looking thing, is in aquatic environments. The eggs develop into larvae that form inside a paratenic host, a second host, which in this case is mosquitoes. The mosquitoes emerge from the water carrying the cyst of the hairworm, get eaten by the beetle, and then the hairworm apparently, based on what we know with crickets and other insects, affects the behavior of the beetle, we think. We don't know this for sure, but other species suggest it affects their behavior. They run to water, and the worm pops out. Our traps in the north had liquid in them. Okay, so the reason that the hairworms were coming out of the beetles was because they landed in some fluids, the beetle did, and then the hairworm came out. So why am I telling you this story? Well, this has all kinds of implications for Arctic food webs because we didn't know these kinds of beetles were eating mosquitoes. And mosquitoes in the Arctic spend their time in aquatic habitats. Okay, the larvae of mosquitoes are in still water habitats. They emerge and the mosquitoes fly around, take a blood meal, the females will take a blood meal, etc. But these beetles are somehow eating those mosquitoes, which is an interaction that we hadn't put together before. The beetles are ground dwelling. The mosquitoes are not. So what do you speculate? Are the beetles jumping off the ground and taking down mosquitoes? Any other ideas?
They're hiring spiders to create webs.
They're hiring spiders to create webs. I like that. So that's one idea. Someone raised that idea to me previously. What I think might actually be happening, so they may actually be, mosquitoes will often land on the tundra as well, so they might be taking them when they land. The other idea is that maybe that the beetles are actually scavenging on a lot of dead mosquitoes as well. So the parasite would still be in the mosquito host even if it died. But somehow there's a connection between an aquatic habitat and the terrestrial habitat, and that clue comes from this hairworm parasite. Okay, so by investigating that story, we're learning more about food web ecology in the north, ecosystem ecology, behavior, the behavior of the beetle when it's parasitized. Behavioral ecology, I mentioned that. Evolutionary biology, we don't know what this hairworm species is. We don't know how it's related to other hairworm species. We have no idea. And biogeography, so where is this thing found around the world? That's my first story for you. The second of three will take us to spiders again. Around the same time I was getting involved with that work, that big project I told you about, we teamed up with some colleagues, some mammalogists and natural resource scientists. Murray Humphreys is his name, some of you might know him. He had an ongoing project along the Dempster Highway, which runs from essentially Dawson City up to Inuvik in the Yukon. It's a splendid place to work because it crosses different habitats from boreal forest in the south end, and then its highest point is actually at Tombstone Mountains, which is towards the south as well. And then it starts to decline in elevation as you move further north, and you start to get, and you kind of get this pattern, they call it a Swiss cheese pattern, when you've got boreal forest with little pockets of tundra, and as you move further north, you have tundra with little pockets of boreal forest. So it's a really interesting for a biologist in general. So we teamed up. It's also, by the way, the most spectacular place on the planet, just as an aside. It's pretty nice. This is my favorite place. It's actually one of my favorite places. It's actually about 10 kilometers south of the Northwest Territory Yukon border. If you're standing to my left or to the left is the Richardson Mountains. To the right is the most spectacular place in the world where I have like 500 pictures, and I'll show you one at the very end. I always stop the car, and all the students are like, not again, because I hop out of the car and take pictures of the same thing every time. It's a beautiful spot. But we can basically go 100 feet off the road and collect spiders there. We don't have to fly in to remote places. The spiders are all over the place. So we use the Dempster Highway as a site for doing all kinds of interesting work. And I went up there with my technician, my grad student, Joe Bowden, and my dad. He came along for fun one year. He was telling me how great it was having coffee with his retired friends afterwards and talking about chasing spiders in the Arctic. It's not your typical retired guy coffee shop talk. And our friend Glenn Fiddick, because the friend Glenn Fiddick always comes out at the Arctic Circle. That's just sort of standard practice. And we studied the spiders in the Yukon Territory. We did some interesting science. It was a lot of fun. We spent a lot of time doing this. And all the tourists would come over and ask us if we've seen moose. And I would say, no, we're never looking up. All we're doing is looking down, collecting spiders and other critters. So we do miss some of the bigger wildlife. We see the better wildlife, actually. Okay. Wolf spiders average more than 1 spider per square meter. That's a lot of spider. If we add other spiders, our latest estimate is, I think, 9.7 spiders per square meter in the Arctic. And don't forget, it's permafrost about that far down. So the square meter I'm talking about is like this table of tundra. That's how many spiders per square meter are in the Arctic system. It's unbelievable. How do we figure this out? 2 ways. One is we took an empty bucket, like a bucket with the hole cut out of the bottom, and planted it down. Did that a lot and counted. You do that over and over and over and over again, you get a density estimate. Very labor intensive, very kind of annoying science, but that's one way we figured out density. The other way is I had a student that bought meditation tents for doing yoga in. The meditation tent is, I should have brought a picture, I'm so sorry. You're supposed to sit in it, and it's supposed to keep the bugs out. So it has a leg hole in it as well. You sit in there, and it's 1 square meter by 1 square meter. So we cut the bottom off the meditation tent, put it on the tundra, dug it down to the permafrost, and then the stuff that was living in there emerged, and we collected in there to get a density estimate. So we were using it for the bugs inside, where the philosophy of the yoga meditation tent is to keep the bugs out. It worked very well. So we have very accurate estimates of density of these things. I find that, is that interesting to you, that many spiders? In the Arctic.
Can you compare that with how many spiders there might be here?
Oh, there's as many here. Actually, I'm not sure there are, because the tundra habitat is very three-dimensional. And so the litter-dwelling fauna here is very diverse, but it's maybe not as dense as up there, because we have the leaf layer, and then we get into the organic layer, and then we get into more of a clay soil underneath. This is really rich. The whole tundra habitat is very, very rich. So it's hard to know. I haven't replicated this in the south. I've done a little bit of work, and some of the numbers are comparable, but I'm not sure. It's a good question. I'm not sure. It'll be similar. There's a lot of spiders everywhere. One of the benefits of wolf spiders are that the females carry their egg cases attached to their back end, to their spinneret. So they're carrying all their babies with them all the time. So as a biologist interested in spiders, this is an incredible opportunity, because you now know what we could call the clutch size, or how fecund, how many babies is that spider going to have. So as a population ecologist, that becomes interesting, because you can get those data very, very quickly. So Joe was working up there for a whole summer, my student Joe, and he wanted to do a project to understand the basic life history of some of the wolf spiders up there. And he was spending a lot of time pulling open egg cases and counting little spiders. I did not do that. A lot of time counting little spiders. But now and then he would pull open an egg case and find this. That does not look like a spider. No, that's not a spider. What does it look like to you? Oh, you guys are good. Wasps, yes, not ants. Wasps. Wingless wasps, but they're wasps. And this is what else you see. That's the egg case. This is like a sci-fi movie, isn't it? Alien. The wasp, that's a great picture of the wasp emerging from the egg case. So Joe also had one of those moments where he was like, huh, what's going on here? So he started counting them. So in some cases, it ends up being a species in the genus Gellus. We have no idea what species of wasp it is. We know that they are egg parasites of wolf spiders. So he started counting those in addition to counting baby spiders. And we wrote a paper about it. And I've highlighted what I think is the interesting part. The percentage of parasitized egg sacs we report are the highest on record and are about 1.5 times greater than the highest recorded in other populations of wolf spiders. And if you look at the very middle of that graph where it says Ogilvy and you look right to 51.50, that's the percent of spider egg sacs that were parasitized. That's over half of the female spiders carrying eggs would not have spiderlings and would instead be carrying around wasps. Half. So from a population dynamics perspective, that's an incredible mortality factor long term, right? You've got a population where half the population is being parasitized by a wasp. That's kind of amazing as well. And I'm not just saying that because I like the system and I find it interesting. Those are really high, really high rates of parasitism. Very unusual. You never see these wasps around, but you sometimes see these little ant-like things wandering around. Mostly it's wingless forms that are there, and I think what they're doing is they're crawling around the tundra, the wasp, and then a spider, and how dense are they? Quite dense. Walk. The wasp must just kind of grab on, pull itself up, and then lay its egg in the egg sac of the spider, and then those wasp babies hatch and eat all the spiders, have a wonderful protected home, and then emerge when they're ready to be adults. It's pretty interesting. And it's, 50% parasitism suggests that this is a very important dynamic in these Arctic systems. So not only are the spiders important at that level of that density, you've also got this parasitism factor happening. That's one of the winged ones. They're very beautiful. Completely, probably new to science. No one has a clue. We connected with the expert on these wasps, and his response was, oh yeah, another one of those. There's so many undescribed species, we don't even know how to start. We just know there's a lot of them out there. Great. So what do we learn? Wolf spider egg sac parasitism helps us understand population ecology, behavioral ecology, biogeography, evolutionary biology. That little story I told you is a whole research program, right? whole suit we were talking about neat ideas the other day, right? Neat ideas to discover for grad students, for future scientists. Go to the Arctic and figure out what's happening with these spiders. Unfortunately, it may not be really widespread. I feel really bad, but I connected with my colleagues in Denmark who hired a student to work and go through 20 years of wolf spider collections from Greenland to look for wolf spider parasites, and she found nothing. Nothing. So they don't seem to be in Greenland. I felt really bad. She spent a lot of time coming up with nothing. Okay, the third of the 3 stories is this one's about pseudoscorpions. Pseudoscorpions are arachnids. They're a very unusual order of arachnids. There's only about, I think, just under 4,000 described species in the world. We don't know how many in Canada. There's maybe 20 or 30 species in Canada. The largest pseudoscorpion on the planet is 7 millimeters long, so they're pretty small. And they look like a scorpion without a tail, like a pseudoscorpion. They kind of live in different sorts of habitats. They are predators. They're pretty interesting, completely understudied. There's only about, I was at you guys would have loved this, the International Congress of Arachnology last summer. The largest ever gathering of arachnologists in the history of the planet, 364 arachnologists at one place. And the entire research pseudoscorpionologists met for a photo. Everyone working on them got together, 9 people in the photo. Wow! The whole group was there. Very exciting. It was, because you never get together with these people. The main guys works in Perth, Australia. There's a guy in France. There's like all over the place. And there's only a couple people that are actually employed to work on these things. Most people are amateurs. They don't attract a lot of research dollars. Okay, I got to speed up a little bit. So I was doing a project on pseudoscorpions during my sabbatical, and I came across a paper by a guy named William Muchmore who said, there's a new pseudoscorpion from Arctic Canada, Wyochernys arcticus, described from Sheep Creek. And then as we were driving up the Dawson, or the Dempster Highway, we came by a sign, and it said Sheep Creek, so we slammed on the brakes, and I was like, wait a second, that's got to be the same Sheep Creek. Has to be. So we all trundled out of the car, put on our bug jackets, and the original description said, under rocks next to Sheep Creek. And they were collected by someone looking for mites who happened to find 4 pseudoscorpions under a rock next to Sheep Creek, next to the Dempster Highway, back in the 80s. Stuck them in a vial, sent them to Bill Muchmore. He's like, it's a new species. He described it, and then just kind of dumb luck, as we were driving by, I noticed a sign for Sheep Creek. We slammed on the brakes, got down there, and look what we found. Isn't that beautiful? Yeah, it is. It's incredible. And look, there you can see the babies, right? She's carrying the brood sac underneath. Absolutely gorgeous pseudoscorpion. So it was there. So what do you do? Well, next time you come to another creek, maybe it's Grizzly Creek, you slam on the brakes, you get out, you send Jean-Francois to go look. It was raining that day. Go look. You can always look for pseudoscorpions in the rain, we discovered. And thankfully, we had lots of rain that trip. So we were able to study under a lot of rocks at a lot of different creeks. And lo and behold, they're all over the place. All those dots, there's Sheep Creek. The Sheep Creek is above the Arctic Circle. Those are the other spots. And we looked down by Dawson. We looked down by Whitehorse. We looked further north. We couldn't find them anywhere else except those dots. We've since added a few more dots, but they don't seem to be anywhere else. This strange Arctic pseudoscorpion. Why is that? I'll come to that. It's a good question. Hold that thought. Aren't they cute? Look at the little proto-nymph. Now you can say you're one of the very few people on the planet that's looked at a proto-nymph of a pseudoscorpion. But I took some data on it. I collected a bunch of them, so we took some data. We measured things, collected some data, and thought about basic. Nobody knew anything about this species other than that 4 specimens were collected under rocks back in the 80s. So we're adding more knowledge about pseudoscorpions. But much more made a mistake. And in 1996, he wrote a paper that said, wait a second, I made a mistake. It's not Weilkernes arcticus. I'm sorry, it was originally described as Weilkernes asiaticus, based on specimens from Siberia, Mongolia, possibly Tibet, and also now Sheep Creek, Yukon. Okay? So there was a taxonomic error that was uncovered some years after Muchmore's original paper. I'm getting to that why. Okay? Why is this interesting? Well, this is the global distribution of the species. I have a friend in Alaska who sent me specimens, and it's the same thing. This is why. The last ice age, there was a section of northwestern North America that's known as Beringia that was not glaciated in the last ice age. We tend to think of woolly mammoths and other exotic creatures that lived in Beringia and wandered across the Bering Land Bridge. We don't often think of the pseudoscorpions as being relics of that time period that are still hanging around under there. So they're a Beringian species that is restricted, we think, largely because they're only living in unglaciated parts of North America over the recent geological past. Very, very unusual. And it's not, these are what we usually envision. We don't usually think about pseudoscorpions, right? Well, hopefully you all read the Yukon News in 2015. My goodness, there was a big story up there. Tiny Ice Age relic lurks along the Dempster. Okay? People are interested. I love this. The local news were really keen to hear the story of this pseudoscorpion because it was so weird, so out there, so unusual, part of their land, part of beside creeks in the Yukon. Okay? Completely accidental discovery in the 80s, and complete accidental that we happened to see the sign on Sheep Creek as we drove by it. That's not how we think of science working, by accident, but I think it works by accident more often than not. Okay? We found it at the top of these little creeks that were only this wide. How did it get up to the top of these creeks? Any ideas? They're very, very slow. They kind of move around a rock, but they certainly don't have long-distance travel capabilities. They would wash down whenever there was rain. They would surely wash down a river, but how did they get back up to the very headwaters? Because they were there in high density. We think this way. They're very good at being phoretic. They grab on to other critters, they hitchhike. So we assume that they grab on to other animals, other insects, that presumably take them upstream again after they get washed down. We have no idea, though. This is a guess. Here's another question for you. This is Sheep Creek, the infamous Sheep Creek. You can guess that it floods every year, quite significantly. The pseudoscorpions were never found right next to the creek, but a little further up, they were found in that area. They were never found when there was real shrub or tree development. So there was a zone that we could find them in. But in that zone, they would absolutely flood every spring. Absolutely. And this, from the very first days of collecting these pseudoscorpions, we're always puzzled around what the heck did they do every spring when those creeks flooded? How did they get through? How did they survive? We know some arachnids are able to breathe for very short periods of time underwater by bringing down oxygen with them like a diving bell. Okay? There's some plastron respiration that we know about. It's not known from pseudoscorpions. Did they just die? Did they crawl up? Well, thankfully, I had collaborators one year, by accident again, in 2015. I happened to be in the Arctic along the Dempster Highway with Brent on the right and his grad student Susan, who was studying wolf spiders to look at cold adaptation in wolf spiders. And we stuck Pseudoscorpions on the research permit, because I always do put the Pseudoscorpions on the permit because there's always more to learn. And while we were up there, we got talking as we were driving. We spent a lot of time driving. Hey, I wonder what those Pseudoscorpions are doing when it floods every spring. And then Susan and Brent are like, well, let's just figure it out. Let's collect some and try to drown them. Okay. I was game for that. Susan did the work, immersed the Pseudoscorpions in oxygenated and deoxygenated water, checked for survival after 1 week and every 1 to 3 days thereafter until 50% of the emerged animals had died. The BBC loved the storyline. 17 days they could be underwater without difficulty. And they were doing that in the deoxygenated water. The Pseudoscorpion group at the Congress I was at, the 9 of them I was telling you about, they had some ideas. Unfortunately, I made the analogy that it was like stomata on plants. you know stomata that open and close? Essentially, the spiracles on Pseudoscorpions have a capability to close. And what we think they're doing is just basically shutting down and waiting until they can breathe again. So they're carrying down, they do have a silvery film around them. They're carrying down a bubble, but they're not using that for respiration. They really are fun to study, Pseudoscorpions. There's a future for you Pseudoscorpion biologists out there. So tiny arachnids tell us about metapopulation ecology, what species are living in which river valley, broader biogeography patterns, life history adaptation, evolutionary biology, and physiology. We can learn a lot from these stories. My lab is basically centered on trying to understand biodiversity in space and time and how it relates to ecosystem function, stability, resilience, etc. I think we need to always insert more natural history into this system, and for all of you working in science, that's my plea to you, is to think through that lens of natural history as much as you can. How do you start? Spend time outside, keep an eye on those old scientific papers. You never know. One line in the paper could change your career, right? An observation, collaborations, always be open to working with people that know a hell of a lot more than you do, okay? I don't know how to put little animals in immersion experiments. That's not what I do. But there are really talented people that know how to do that. So collaborations are key. And I think almost most importantly, curiosity, wonder, and passion. So when you see these little critters, wherever you stumble across them, look through them in that appreciative frame of mind instead of the, whatever, just another thing. It's not just another thing. It has a story to tell. So Joe and Crystal were really foundational to my interest in natural history, and were the sort of people that were open to answering questions that may not be the sort of question that you'd apply for a major grant for, but really interesting and leads to new discoveries, okay? So when we think about what a naturalist is, and many of you are naturalists, I know this. Curious, observant, passionate about the world around you, fascination for nature. Naturalists often document what they see, enjoy sharing knowledge, and motivated to continually expand that knowledge. So whether this is one of my kids collecting seashells, that's a pretty good natural history moment, right? I like this one in particular. This is a guy that lives in my town who just likes trees. He likes them so much that he learns so much about trees, and he goes into the local elementary schools and works with the school to say, can I bring out the kids down to the river valley, and this was in like a mid-January freezing day, and tell stories about trees. But look at all the hands up, right? Trees, yeah, they're interesting. So whether you're young or old, whether you're sitting in an airport watching birds, those are all natural history moments, right? How are we doing at university? This is the sort of grim part. We're not doing so well, okay? The number, this is mainly from US data, the minimum of natural history courses for a BS in biology has been decreasing over time, as has the natural history related material in basic biology textbooks have been decreasing over time. We have to turn this around. We're doing well at McGill, okay? We've been teaching, the course that Val mentioned is natural history field research, and we're writing papers to talk about how to reintegrate natural history into university classes. We can do this, and McGill is a wonderful place to do it because of the talent and because of the in general, a lot of professors at McGill embrace that idea of observation as a very valid form of science. Taking students outside, it's never too early or never too late to take students outside and have a heck of a lot of fun in the pouring rain looking for, crayfish under rocks in St. Anne de Bellevue. Okay? And you can depict the scientific method embedding natural history observations directly. That top, in the project, we use natural history observations as a foundational entry point into the scientific method. And we can do more and more of this. And then I would say also, we've got a blog, listen, speak, write, engage, tweet, share as much as we can, all right? The more we can share our discoveries, whether we're experts or not, experts in the traditional sense, we can become experts and share. And we can watch the robins eat worms. Right? And let's make sure that kid that's watching the robin eat worms that later on is interested in fungi in a forest. Let's make sure that keeps happening, okay? That's a very important piece to not lose that passion. The last little piece I'm going to leave you with. Any ideas what this is? Black fly larvae. Yeah. Black fly larvae. I said that I would bring this back to the Arctic system and to that historical point and the fragile Arctic. One thing we did discover with our project was that over time, the black fly species, different black fly species are moving north. And we relate this directly to changes in temperature. In particular, a species called Simulium tormentor. Just use your imagination. Simulium tormentor is now on Victoria Island, where Cambridge Bay is located. It wasn't. We discovered it in 2006, large work in the Arctic. And the models suggest it's entirely because of temperature. So we are getting advances of black fly species. This is more than just a problem for humans because of this, but I will say it. We're now seeing wildlife affected by things like black flies, in part because of climate change. Okay? So there are real profound effects that are happening because of climate change. And we need to have good understandings of the biology of these species, the identity of them, in order to properly track and understand these changes. We absolutely have to. I promised you the picture of my favorite place. It's not bad, huh? Yeah, so when you think about the fragile Arctic, this is sort of the image I'd like you to have in your mind and treasure, and get there if you can. Study natural history, and it'll be very satisfying, not only for you personally, it'll also help us out in understanding our biodiversity. Thank you. You're welcome.
I think we all have a newfound appreciation
for little creatures.
Of all these little creatures. And even if they're in like 9 of them in a square meter, I feel like I can live with them a little bit more now. So thank you very much.
Thank you. Yeah. Thank you. I know we're pretty much at an hour, but if we have a few minutes for questions, I'd love to have some questions if there's time.
Is 17 days enough to survive the flood? 'Cause 17
Yeah.
days seems like really small.
So we looked at the data on the flood data on some exemplar streams in that part of the Yukon, and it's 10 to 12 days is the estimate. So yeah, and that was 50% were still alive after 17 days. So they'd be fine. Yeah.
As it warms up, are things that used to live in the Arctic conditions, are they moving north too to preserve their environment? Or do they just lose some of the
We don't know well enough yet, and so ideally we need to go back to the sites we sampled and do that very robust sampling protocol in another 5 or 10 years to kind of address some of those northern shifts that might be happening. We don't know right now. What we are seeing is some southern species that are moving further north. That's absolutely. We have data on an Arctic yellow jacket wasp, for example, the black flies, and some other data with a couple other species that suggest that northern shift is happening. We don't know about the flexibility of the northern species relative to changes temperatures in the same way. Good question, though.
So I remember from our classes that we were talking about food webs in the Arctic and saying that they were inverted, so we have more predators than preys. First of all, does that still, like, is it still the case? Will we find something else that does this?
Right.
And if I remember well, it was because of intragular predation, so spiders eating each other. Is there like any new data that says that helps to explain this phenomenon?
So I do have a PhD student now who's been modeling food webs, and the question Val's asking is around when we think of food webs in a textbook, we see this big bar that represents all the grass in the world, and then we see the little rabbit that's a smaller bar eating the grass, and then we see the eagle that's a little tiny bar that's eating the rabbit. Okay, we think of it as a pyramid with a broad base. With a lot of the data we've collected from the Arctic, we don't see such a strong, we see an overabundance of predators, and it's kind of confusing. So one of my students has modeled some of that, and some of the models suggest it might be more truncated as opposed to inverted, but there's certainly lots of predators, and one of the reasons that's not sustainable, having a top-heavy food web, but there's probably a lot more species being omnivores, eating different things, or predators eating predators probably happens a lot. So we do have some new data on it, but it's still an open question in many degrees. Thank you for that. But yeah, it's a great idea to see textbooks not always depicting food webs in the same way, because there are other shapes of food webs.
Any other questions?
Can I ask you, what time of year were you there? Where's the snow?
There must be snow. There's always snow. I've never been there without snow. Most of the work on the spiders that we've done has been focused in July. That tends to be the period when the males and females are active in mating, and that's where we get all the life history data. So most of the work's been in the midsummer for sure. In the very high Arctic, when we had the teams going there, it was a very short 2 or 3 week window, also in the most active season. We've not looked. Things really get quiet by early August, mid-August in there, so it's a very, very short season, and that's when we tend to focus. But I'd love to spend time in the early season, because from what I know from biologists that are there when the snow is melting, the spider activity is very, very intense right as the snow is melting, and that's why we are pretty sure that the spiders are a key food resource for birds that are first arriving, because a lot of the other insects haven't emerged yet, but the spiders are active. That doesn't answer the question, but it's a different answer to a different question. Yeah.
What kind of grants from Walmart?
Pardon me? Walmart. Oh yeah, you noticed the Walmart. Walmart Canada. So Joe was quite enterprising. Joe Bowden needed to find money for doing research. He was working on different proposals, and he happened to call up some guy at Walmart, and the guy he got on the phone was like, "Oh, I really like biodiversity, and yeah, we'll fund you." I was like, "What?" However, his supervisor found out and was like, "No way, we're not funding biodiversity research in the Arctic." And then the guy felt so bad, he gave us a whole bunch of store credit. And so we cleaned out camping supplies in the Whitehorse Walmart one summer. It's really quite a whole funny, someone always notices the Walmart piece in there. It was very, very, very funny. Yeah. So they were supportive in the end. Yeah. The tents were crappy though.
All right, we'll take one last question.
Yes.
Sort of methodology question. When you're traipsing around like that looking for spiders, just actually what do you do? Do you dig them up? Do you put them in little jars? How do you carry them around? When do you look at them there in the ground? You sit and watch what they're doing for a month, or do you take them back to your tent? Or
Sure. So
more about
I'll give you the 30-second version. We often carry an empty yogurt container around with us, and we push the little, get the spider to jump in the container, then we put it in a vial and take it to a makeshift lab, which is a cooking shelter at one of the campgrounds, and do the measurements there. Sometimes we do work in the field with them also, but more rarely. Usually we take them, study them, sometimes put them back, sometimes bring them south, depending on the project. It's very, there's different techniques for collecting spiders in the field. The yogurt container one is one that I particularly like. Other students like other approaches. But yeah, we essentially walk around with vials, and it's a very interesting work. People find us very strange.
and then you mark
Yeah, so we would collect all the metadata associated with the sample technique. For the bigger project, we had traps that would stay out for a longer period of time and do what we call passive collecting, and then go back and check the trap to find the spiders in it. But for some of the life history stuff, we were collecting them live and then often measuring them right away. Yep.
Excellent. Well, thank you all for coming out this evening. If anyone has any more questions, feel free to come find Chris afterwards. I'd like to invite you all to stay for our reception and to come get some ice cream sundaes. So thank you.
Thank you.